From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 33, recorded on August 29th, 2022. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from San Francisco, Ori Lieberman.
Hi, Vincent. How's it going? Good to see you again.
Okay, winding up the summer here, aren't we?
Yeah, it's crazy. I mean, it still feels like, it feels like winter here, but you know.
Also joining us from Salt Lake City, Jason Shepard.
Hey guys, yeah, definitely not feeling like winter over here. This week it's still going to get up to 100 degrees, so it's pretty crazy.
Oh, man.
100?
Yeah.
That's pretty hot. I'm not sure what it is here. I don't have my phone. And from New York, Tim Chung.
Hi everyone. I can tell you because my laptop tells me it is 83 Fahrenheit, but I have no idea what that means because I'm not an American.
It's warm, but not hot.
It's not.
It's pleasant.
Yeah, it's okay. It's not 100.
It's rather humid in these parts. So its humidity is quite high, yeah.
Yeah, there's a storm coming. Once again.
Well, for this, it's not really summer. Summer isn't over until the 20-something of September, right? Something like that.
I don't see the leaves changing.
Yeah, right. That's what does it, right? Well, anyway, for this near-end summer episode, it's my turn to talk about a paper. And, of course, I picked one that has viruses in it because at least I can know something in the paper. But I have to say, I did work on a neurotropic virus most of my career. Actually, multiple neurotropic viruses. So I have thought a lot about viruses in the central nervous system, although this paper tackles a subject I never really thought much about. So I learned a lot. This is a paper in Nature Neuroscience. Title is Meningeal Lymphatic Vessels Mediate Neurotropic Viral Drainage from the Central Nervous System. First two authors, Xiaojing Li and Linlin Qi, or Shi, and Bin Wei is the last author. This comes from a number of institutions throughout China. And, my viruses, we study how they get into the CNS. It's harder to get out. Very few viruses end up getting out. But here we're going to talk about a potential mechanism for that. And this has to do with the meningeal lymphatic vessels, or MLVs, which are just beneath the skull, at the top of the dorsal and ventral part of your brain. And, lymph vessels elsewhere, what they do is they drain tissue fluids. They bring them to the lymph nodes, which have collections of B and T cells, and there, any foreign antigens can be interrogated and so forth, or self-antigens can be interrogated as well. And so this is potentially a system for doing that.
Is it kind of like how in New York we're now kind of surveying the sewage system for any weird new COVID or polio viruses?
That's right. It is a surrogate, right? People always say, well, am I going to get polio from the sewer? No, you're not going to get it from the sewers. It's a surrogate for saying that people are crapping out the virus, right? Although in the sewers in New York, we process sewage and so forth, but we don't make immune responses.
No.
But the lymph system is important for that, to get the antigens into the lymph nodes. And so the brain has this system of lymphatic vessels just beneath the skull, and they have all sorts of their cells that make up these vessels, lymphatic endothelial cells. They also have meningeal LECs. So these are meningeal LECs, and we have conventional LECs, which are elsewhere in the body. They have markers which you can use to distinguish them, cell surface markers, and they use these extensively throughout the paper. And so it's already known that these MLVs carry cerebrospinal fluid components from the CNS to the brain. And remember, the brain is bathed in CSF, and that fluid can flow throughout the brain and also can end up draining into these lymph capillaries, which are very permeable. Lymph capillaries in general are more permeable than blood capillaries, and so components can get into them and be taken to the lymph nodes. And these brain lymph capillaries drain. They coalesce into main vessels that come out of the skull at the base, and then they drain into your cervical lymph nodes, the ones around your neck.
Are those the ones that swell up when I have a cold?
They do.
Yeah.
I don't know if they're the same. Are they the same ones, Ori, that would swell up?
I think so, yeah.
So they're also draining.
These are like submandibular, I feel like, here. And then they're
they're different ones. Oh, okay.
But if they're swollen, it's most likely you have a cold and not a brain infection. Right?
You hope.
So Vincent, these vessels, and I guess you guys can also, Jason and Tim can add to this. They feel like they were really only recently discovered and characterized. And
yes,
for a long time,
2015, only 7 years ago.
For a long time, there wasn't thought to be lymphatic drainage from the CNS, and then work by Yoni Kipnis and others have shown, kind of characterized these vessels and then shown that their dysfunction or their function plays a role in many different neurologic diseases, especially neurodegenerative diseases, and that they're really important, for example, for draining toxic proteins that we know build up in neurodegenerative diseases. Whether or not their buildup causes the disease is a separate question, but at least are important for draining those proteins.
Yeah, I mean, this is one of those prime examples in science where there was dogma based on not a lot of data. And it kind of goes back to this idea that the brain was immune privileged and there was no interaction between the immune system and the brain under normal circumstances. And it was literally like, oh, well, we'll just go. No one's actually looked for these things properly. And when they discovered that there was this whole system there, that sort of opened up this whole idea that there is this close contact between, fluids both in and out of the brain that could have a real impact because of the immune cells. They've subsequently discovered that there's a bunch of immune cells there that can then interact with secreted molecules from the brain and vice versa. They secrete stuff, and that also impacts. I mean, Yoni Kipnis has even shown that it impacts normal cognition, which is pretty crazy.
Most virologists study how viruses get into the brain, right? Which is very well protected. Not a lot of viruses get in, but some do. And once they get in there, they can cause a lot of trouble. There's the so-called blood-brain barrier. We have astrocytes which wrap around the brain capillaries, and they're very good at protecting the brain. But some viruses can get around them. There's also, of course, the olfactory nerves, the trigeminal nerves that can let herpes viruses into the brain sometimes. And also some can enter via the cerebrospinal fluid. So some viruses can infect the ependymal cells, which are near the CSF, and which are, the generating cells of the CSF. And they can get into the CSF and then travel extensively throughout the brain. But nobody has, and this is a reasonable question, nobody's addressed whether these meningeal lymphatic vessels could help viruses get out of the brain. And as they're pretty recent discoveries, so it's not surprising. But that's what they do in this paper. What happens to meningeal lymphatic vessels, MLVs, after infection? So they use mice, and they use a variety of viruses that can infect mice, including rabies virus, Zika virus, herpes simplex 1, Japanese encephalitis virus, and vesicular stomatitis virus. So these are all neurotropic viruses that can infect the brain. And remember, though, these are mouse models. So, we don't know the complete applicability of what we find to humans, because that's what a model is. It's the best you can do, of course, and then you make some observations, and hopefully you can go into people and make observations in the absence of infecting people, of course.
I just want to clarify, these are in different mice that they got infected, these viruses. They didn't put all these viruses in one mouse.
No.
But this is I didn't realize there's so many different viruses that can all go into the brain. And then in the paper, they mentioned they infect via different routes as well. So some of the virus, they actually inject into the muscles. Some is directly into the brain. Some is into the veins. and then the herpes virus is into the eye, which is I didn't know you can get.
These are all models where we have learned how to put the viruses in, because they don't always get in by the same route. And again, it's a model because, for many of these, that's not how people are infected, right? So you don't have to inject herpes virus. You don't have to put herpes virus into the eye. You typically get it from saliva from your mother or father, and it would go into your mouth. But that's what an animal model is. And yes, each time they infect the animals separately. So the first question, can virus infection induce lymphangiogenesis? In other words, proliferation of these the cells that make up these MLVs. So they take 5-week-old mice. This time they intravenously infect them with Japanese encephalitis virus. And what that does in mice, this is a human virus that is spread by mosquitoes, but in mice are susceptible, and they get encephalitis. They get inflammation of the brain. And they can immunostain with antibodies against various markers of these lymph capillary cells. And they can see expansions of the vesicles in different parts of the brain. They can see increased diameter of the cap of the lymph vessels as well. So this is after intracerebral inoculation.
The pictures are really striking.
Yes, this is one aspect of this paper that's amazing, is that they have lovely visuals. They have a variety of techniques that they're going to use of increasing sophistication, and the images are beautiful.
Yeah, but I didn't realize the lymphatic vessels can increase by so much after infection.
Yes.
It's kind of similar. It reminds me of supposedly if you have a wound like in your body, then your blood vessels will undergo angiogenesis to try to close the wound, and a lot of blood vessels would form around it. And this is kind of reminding me of something like that, but for lymphatic vessels. Yeah. And I didn't realize that can happen. Yeah.
I did not also. I presume it's a response to infection, right? Because that's
right.
It's not that the virus is inducing this, but the host wants to do this in order to try and do more sensing. So intracerebral JEV, intravenous JEV. It also happens when you put the virus directly in the brain, intracranial inoculation. So for many viruses, if you put them peripherally, they never make it to the brain. And so you put it directly in the brain, which is, albeit very abnormal, right? That's not how we get our viruses or any animal, but sometimes you have to put virus directly in the brain. I spent many years putting poliovirus directly in the brains of mice. You put a small volume, typically, I used to put 50 microliters in, which was a lot. But what's amazing to me is that they recover from anesthesia and they're running around like nothing happened to them. You just put this huge needle in their brain and they're fine.
We don't need our brains.
Yeah, we put big cover slips on top of the brain to image into the brain, and they recover from surgery very quickly.
Amazing.
Mice are really pretty amazing. They're sort of normal even after craniotomies.
So you put Japanese encephalitis intracranially, you see the same proliferation and increase in vessel diameter. At the same time, the virus is reproducing in the mice. They can measure viral levels in the brain. They can also measure disease severity, different kinds of behavior alterations induced by encephalitis. And so it happens no matter how you put it in. And then this increase in the diameter of the vessels, they say, happens about 3 days after infection with Japanese encephalitis virus. Okay, so that's meningeal lymphangiogenesis. They also look at other viruses. They infect mice with vesicular stomatitis virus, and this they do intranasally because it was found years ago that if you do that, you put the virus in the nose, it gets it into the brain, presumably via the olfactory bulb, and it reproduces there, so you don't have to stick it right in the brain. When you do that, the mice get brain infection. You can find brain in the CNS, and the mice lose body weight. And they also see the lymphangiogenesis in these meningeal lymphoid vessels. They use herpes simplex. They do corneal infection with that virus. Again, it invades the brain. The virus reproduces in the brain. You get a loss in body weight. And again, the lymphangiogenesis is also observed. They also did rabies virus.
Vincent, I was just going to ask you a question. Do you think that this would only happen with neurotropic viruses, or do you think that this is just a general inflammatory response to a systemic infection, for example?
I suspect that if the virus got into the brain, it could do similar things, right?
But you think it requires CNS infection? That this isn't happening every time you have a common cold where there's no viral?
I don't think so. I would think not. Yeah, I would think not, because I don't think that virus is getting in there and it's not reproducing enough to trigger by whatever mechanism, the lymphangiogenesis. So yeah, I think this is very specific, most likely. Although I know we used to put rhinovirus in the brain of mice and absolutely nothing happened. So that would have been a good control.
Right.
Yeah. But I quickly looked up whether if you have a peripheral infection, whether peripheral lymph vessels also undergo lymphogenesis, and they do apparently. So it's not just
makes sense, yeah,
in the brain.
Yeah. They also looked at rabies virus, where you can inoculate mice intramuscularly. You get high lethality, and the mice get paralyzed at 8 days after infection. Again, increased vessel diameter. Proliferation of the lymph cells.
I wonder what safety these researchers have to undergo when they're handling rabid mice.
Jason would likely know, because you may use rabies to target synaptically, right? In some cases.
Right. Yeah. I mean, we've sort of modified some of these viruses to do tracings, because we know that rabies for sure jumps synapses, and so we've used them to modify it so we can actually trace where circuits are in the brain. But
you have to be vaccinated, right?
But these are, we've taken the bad things out of the virus. So even if we did jab ourselves, we would hopefully not get an infection.
But in this case, they're using, street rabies. So you have to be vaccinated. If you're vaccinated, then you'll be fine.
Okay. I always wondered if there are like mice model of rabies, because rabies causes one very, very odd thing that I've never read anywhere that anyone has figured out, which is this thing called hydrophobia.
Yeah.
And normally you think hydrophobia, you mean like grease, like in chemistry. But this is like in humans, if you have rabies, you like really run away from water. And
I don't know.
Neuroscience-wise, that's fascinating. How do you... Wow. No one has ever has a normal phobia of water except for in rabies. So I wonder if mice also, if you put some water in front of them, they would freak out.
I'm not aware of that. I think they get such lethal disease and paralysis that they're not running from anything.
Yeah.
All right. So then, so basically what they find with Japanese encephalitis, VSV, herpes, rabies, Zika. Actually, they also looked at Zika virus. I didn't mention that. They all induce lymphangiogenesis of the dorsal MLVs, so the ones under the top of the skull. And so then they look at basal MLVs, and they also have similar findings, right? By again looking at these markers and so forth. They also take some human LECs, and they treat them, they infect them with the Japanese encephalitis virus in culture. And they see an increased expression of these markers that they've been using to trace lymphangiogenesis in mice. So that's a nice experiment that they, it's about the best you can do at this point, right? The virus is increasing proliferation in human cells as well.
I mean, maybe we should also note with these whole mounts. So basically with the mice, right, they're taking the whole brain and they're using this technique to clear the brain and literally clear it. So there's this cool image there of a brain before and after they've cleared it. When you clear it, it looks like it's invisible. And essentially over the last 10 years or so, there've been these breakthroughs in clearing tissue. So essentially, getting the pigments out of the tissue so we can put antibodies in there and you basically image the whole brain at once. And it's pretty remarkable that but the way they do it is they make a gel. So they turn the brain or tissue into a gel, and then they extract all the lipids so that and that's essentially what clears it, leaving the proteins behind. But it's pretty cool. And this technique they called iDisco, but there's various versions of it where they essentially just remove the lipids and clear the brain.
Yeah, I found a pretty cool video on YouTube I can put in the show notes of a cleared mouse.
But I mean, this picture is so striking. I mean, you literally can't see anything.
Yeah.
Making even bones transparent is pretty incredible.
Yeah, so that they do a series of experiments which are basically 3D imaging after infection, so that you can see the actual structure of the vessels. And what they say is you can still see them attached to the skull cap, right? So the vessels are still there, and again you can see by these various imaging, including iDisco and other 3D imaging, that the virus infection promotes lymphangiogenesis. Okay, you can see it in a three-dimensional fashion. One of the proteins that increases in the brains of virus-infected mice is VEGF-C, vascular endothelial growth factor C, this is one of the growth factors that's making the lymphangiogenesis occur, and they want to know where that's coming from. And apparently, neurons, astrocytes, and a certain subset of monocytes and macrophages are secreting VEGF-C after infection, which then promotes the lymphangiogenesis in the brain during infection.
Yeah, I think the receptors are on the vessel cells, right?
That's right.
Lymphatic endothelial cells, and they receive these signals and I'm guessing they start dividing.
Yes, exactly. So they ask next if these new vessels are functional. Are they actually working? So they do some, they inject some radioactive tracer called Evans blue. They put it in the intracisterna magna of mice, which is at the base of the skull, right? And this is basically like putting it in the CSF. I mean, I think, and correct me if I'm right, Ori, that sometimes you could do a spinal tap there instead of in the spinal cord, right?
Yeah, I would be very scared to get that as a patient, but yes.
Yeah, so they can inject a dye into that in mice, and this ends up in the CSF, what they see, then they do a time course in either virus-infected or uninfected mice. And they look for this dye in the dorsal cervical lymph nodes, right? So the idea is you put it in the CSF. The CSF is draining into the lymph system under the skull, and then that drains into the cervical lymph nodes. So if you find the dye there, it means that you're getting it there. So in control mice, you can find it there after injection in the cervical lymph nodes. But if you infect the mice, you have actually an inhibition of drainage of this dye into the cervical lymph nodes. So the virus infection is somehow promoting an inhibition of draining into these lymph nodes.
And I think just to, people might wonder, how do you know that it is, when you inject this dye into the CSF in the brain, how do you know it's getting into the lymph node via these vessels, lymphatic vessels we're talking about? People have previously actually physically done the experiment where they physically tie up these vessels with tiny little threads to make sure nothing gets past. And in that case, if you inject the dye, then it doesn't go to the cervical lymph node. So that's how they know it must be going through these vessels. Yeah,
which is an experiment they actually do in this paper as well with viruses. Yeah. They also do real-time fluorescence optical imaging to monitor the movement of materials after injection into the intracisterna magna. They use a fluorescent dye conjugated to ovalbumin. They inject this, and then they can monitor the movement of this after either in infected mice or in uninfected mice. And in uninfected mice, you can find this dye in cervical regions of control mice within 30 minutes or so. But in mice infected with Japanese encephalitis virus, you see very little fluorescent dye there. So again, the somehow virus infection is impairing the movement of material through this lymphatic system from the brain to the cervical lymph nodes.
It's actually really counterintuitive because, previously, they saw that when the mouse is infected, you have double the amount of these lymph vessels whose job is to transport things into the lymph node. And then the second part of the experiment, they show that even though they have, twice the capacity or, many more capacity of these lymph vessels, they're actually not working. Things are getting blocked up for whatever reasons.
Can you imagine doing that experiment? Like you do the microscopy, you see more lymphatic vessels, and then you measure the flow, and you're expecting an increase in flow, and then you're like, "Oh crap, got to figure that one out."
Yeah, exactly. That must.
So they actually compare the two. They would say when meningeal lymphangiogenesis appeared at 3 days post-infection with Japanese encephalitis, right? There's no difference in the fluorescent signal in the cervical region or in elsewhere after infected mice. And at 4 and 5 days after infection, the ovalbumin fluorescence was lower than in control group. So there's an active inhibition there. And it doesn't make a lot of sense, right? Unless it's a viral mechanism to prevent being sensed, in which case I've never heard of that at that level before. That would be quite remarkable, right?
I don't know. I probably would ask this question. They're going to get into the mechanism of what might be causing this. So I think I'll have a weird question coming up. Sorry.
So if they put virus in the nasal mucosa, they don't find signal. If they inject the fluorescent ovalbumin into the nasal mucosa, they don't find it in these cervical lymph nodes. So this is very interesting. So what's ending up in the cervical lymph node is draining from the lymph system in the brain, in the inside of the skull that we're talking about. So they say that the MLVs, not nasal mucosal lymphatic vesicles, represent the primary route for drainage of soluble constituents into the cervical lymph nodes. So then they look at herpes simplex virus after corneal infection to again track the drainage of molecules in the recovery phase. So they put herpes simplex virus in, and some of them die, and then they begin to recover, they gain weight, and so forth. And they say during the acute phase of infection, the fluorescence intensity, so again, we're injecting this fluorescent ovalbumin into the CSF and looking at them in cervical lymph nodes. During the acute phase, the fluorescence intensities are lower than in control mice, but once the mice recover, then there's no difference between the virus-infected mice and the control mice. So they say this suggests that the lymphangiogenesis is induced during the acute phase and then returns to homeostasis during the recovery phase.
I was wondering, because herpes virus is, from all the TWiV that I've been listening to, herpes virus are famous because after the acute phase, they can go latent. They can hide inside your nerve cells. Yeah. So is this perhaps a model of herpes virus going latent and showing that these mice, the lymphatic vessels shrink back even though the virus is latent? So
this is an interesting question. So what they're looking at here is an acute model. So it's actually a scarification of the eye that you use to get the virus to go into the brain. And the virus in the brain is a rare outcome of human infection. It's like 1 in 200,000 people have the virus goes into the brain. It shouldn't be going that way. It should be going from the mucosa to peripheral nerves, but sometimes it goes the wrong way. In mice, a certain fraction of those mice will have latent infection, and you can reactivate them you actually have to put them in a water bath and heat them up to reactivate the virus. It's a weird model, very weird model that some people don't like at all. But so yes, you could study reactivation from latency, but this is an acute model.
I see. So even like other viruses that is in the herpes class, like Epstein-Barr virus that might be linked to like multiple sclerosis, those don't actually go in the brain. They are like in the peripheral nerves.
No, though Epstein-Barr virus is a B cell virus. It infects B cells and goes latent in B cells.
Oh, it's in B cells. Okay.
Yes. Now, varicella-zoster virus is a neurotropic virus that, again, becomes latent in peripheral nerves, not the central nervous system. So when herpes gets in the CNS, it's an accident because usually it's in peripheral nerves. Yeah.
Okay.
They also... They have another experiment where they use fluorescent nanoparticles. These are called Q dots, right? You can get these nanoparticles that have different fluorescent molecules on them, so they'll fluoresce at different colors with different wavelengths of UV light, and they're particulate, so they're dots. You can see the dots when you.
I think Q stands for quantum, just to make
quantum dots. That's right. They're quantum dots. And they can inject these and look, they do this thinning of the skull, which we mentioned before. And then you can do 2-photon fluorescence microscopy where you can actually look through relatively thick layers of tissue, because normally you can't see very far. But the way 2-photon microscopy is set up, you can go, is it like a millimeter or so through tissue to see the fluorescent signal. So Q dots, 2-photon fluorescent microscopy. I remember, Ori, years ago, Richard Axel got the first 2-photon microscope at Columbia. He was so excited about it. He used to pull people out of the elevator, say, "Look, this is our 2-photon." It probably cost $1 million, at the time.
Yeah, I think my PhD advisor got one in like the mid 2000s, and he like it was $1 million that he raised from a private donor, essentially. Yeah.
Yeah, I mean, we've got one in my lab, just for my lab. And
wow.
I remember, they're expensive. I negotiated to have one for my startup. I think now they're only about 300,000 at the most. But I remember coming from New Zealand, the whole of New Zealand, which is, there's only 4 or 5 universities, but the whole of New Zealand had one 2-photon in the whole country.
Yeah, so, Dave's lab has 4 lasers, I think. So
yeah,
wow,
I remember seeing Carl Savoto, who built his own from scratch, would have like 10 just lying around.
Yeah.
Anyways, so they use this to look at particle drainage. These Q dots, kind of a similar experiment as we were looking at, but a little more sensitive, and so they see that as a clear delay of particle Q dot drainage in virus-infected mice from the brain to the cervical lymph nodes. And so basically they say, so these vessels, these meningeal lymphatic vessels expand after virus infection, but they lose their function. They're not able to drain components from the CNS to the cervical lymph nodes. Next, they want to look at the mechanism for this expansion. And so we know, for example, that an increase in lymph flow needs muscle cells to help contract the lymph vessels. There are valves in the lymphatic vessels that regulate the flow of lymph in one direction or another, so it doesn't go back into the CNS, for example. And of course, the proteins in the muscle cells and the valves are regulated by transcription factors, and this has been pretty well studied. So they look at these proteins that are known to regulate the muscle cell and the valve development. And basically, they find that when you infect the mice with these various viruses, the infection reduces the production of transcription factors that are critical for valve function and smooth muscle recruitment. So that's basically what they think is the source of the dysfunctional flow in these virus-infected brains, that the virus infection is somehow impairing the proper muscle cells and the proper valves in the lymph vessels that are needed to get that unidirectional flow.
But I wonder, these cells have also recently just been born because of the lymphangiogenesis. So maybe when they're recently born, they are just not making these valves.
It could be, yeah. Maybe it's a example of, yeah, you made a lot of them, but you made crappy ones, right?
I can just imagine the lymph, like there's these new tunnels that have been made, and the lymph is just circulating around one of them, like just going round and round and round. And
they also find that infection, at least with Japanese encephalitis, increases apoptosis of these lymphatic cells. So that's also probably contributing to the dysfunction of them. Okay, so as we said at the beginning, these vessels have a role in mediating cerebrospinal fluid drainage in a variety of situations. So they say, yes, go ahead.
Vincent, before we move on, I got a question that I was going to ask earlier. So they are trying to link these poor drainage to cell pathology, like cell death, and these endothelial cells not working as well. But I'm just wondering, in all these cases, there is encephalitis, so there is inflammation in the brain, and I wonder if the brain might potentially swell up, because I remember listening to TWiN episode 1 a long, long time ago, where you and Dixon talked about West Nile virus.
That's right.
This is a long time. Like Ori told me about this like 5 years ago. I remember. You and Dixon talked about West Nile virus, and Dixon raised this point. I might have remembered incorrectly, but it stuck in my head because it's an interesting point. Dixon mentioned that in West Nile virus, you can get brain swelling from the encephalitis, and in fact, babies have a better survival rate in this case because they have a soft spot in the skull where the skull hasn't closed yet, called the fontanelle, not fentanyl. I always get those two confused. There's a soft spot where the skull hasn't closed yet, and that can relieve some of the pressure. So I'm wondering, if there's swelling of the brain, you can imagine it pressing against the lymph vessels and blocking kind of drainage.
That could be another aspect of it.
Yeah, they didn't talk about measuring the pressure of anything. So yeah.
No, in fact, in many brain infections with viruses, the brain swells because of inflammation, and that's the problem, right, Ori? The brain is swelling, and that can cause damage.
Yeah, in addition to all of like the cytotoxic and neurotoxic damage that happens from the cytokines themselves, right?
And so is my understanding that in some cases you can treat it with steroids, right?
Yeah, especially like brain swelling, we usually empirically will treat with steroids if it's cytotoxic edema.
So in that episode, Tim Dixon mentioned that sometimes you can treat West Nile encephalitis with steroids. And then Michael Diamond, who was at WashU, he visited years later, and I told him, they said, oh, no. That's the one encephalitis you do not treat with steroids. It doesn't work. Dixon didn't know that. Neither did I. Anyway. Okay, so then they want to look at whether virus particles go from the CSF to these cervical lymph nodes. So could this be a route of viruses getting out of the brain? And for me, this is a very important question because my entire career, I've assumed that there's no virus getting out of the brain. It gets in, but there's no mechanism for it to get out because these lymph vessels weren't known for many years. But it kind of changes the landscape. A virus can get out of the brain and infect elsewhere again and so forth. So they put Japanese encephalitis into the brain intracerebrally, and they use transmission electron microscopy. They do immunostaining to look for viruses in the cervical lymph nodes after putting them in the brain, right? So they're going to drain from the CSF through this lymph system to the cervical lymph nodes. And they see them there. They see particles there, and they see viral protein by immunostaining. But they don't find viruses or viral proteins in the axillary lymph nodes, right? The armpit lymph nodes. And they can they do an experiment where they take out these cervical lymph nodes and homogenize them and infect new mice with them, and they get infected. So there's infectious virus in that. I don't know why they didn't do a plaque assay, frankly.
They did plaque assay. It's in the supplements.
It's in the supplement too?
Yeah, there's plaque assay. There's a plaque assay in Nature News, in this neuroscience paper.
Because that's a pretty, involved experiment to show there's infectious virus there. But there is.
But this is really the ultimate test, right?
It is the ultimate one, right? So the idea is that, yes, these vessels are draining virus out of the CNS, and at least it can go to these cervical lymph nodes. Okay, so now, go ahead.
I thought it might make sense because if I remember listening to Immune, the other podcast about all these things, lymph nodes are important because that's where the germinal center is going to be made. And
exactly.
That's where, I don't know whether you're going to talk about it later on, but that's where I think Rich Condit on Twitter sometimes talks about your B cells going to grad school. Where they are shown the virus, and the B cells have to learn to target the virus better and better by making better and better
so
antibodies.
This is why drainage is important, because it brings. So if in the brain, if a macrophage or a dendritic cell could pick up a virus particle, it would then go through the lymphatics to the lymph node and present it to T cells initially, right? And then if it's foreign, the T cells would start being activated, and eventually they would interact with B cells. So yeah, this is a really important procedure for getting the immune system going, getting the immune response going. Absolutely. So, if indeed these viruses are blocking it, I don't know if they are. I don't think we know that from this paper. It would make sense because it's a kind of antagonism, but I don't think we can say that yet. Then they do the ligation of the vessels. These are tiny things that they're surgically ligating these to these vessels that drain to the cervical lymph nodes. And then they inject virus into the brain or the cisterna magna, and they can see that they by ligating these vessels, you get less virus in the cervical lymph nodes. They detect these by a variety of ways. And so this tells them that indeed the viruses are draining. They look at a variety of their viruses here. The viruses are in fact draining. They also show that you can infect these lymphatic cells with some of these viruses, and they induce apoptosis. So further complicating the situation, apparently, as the viruses are moving through these vessels, they can infect the vessel cells and damage them as well. But I think this shows that viruses can get from the CSF to cervical lymph nodes, and maybe they're even transported in infected cells along the way. But then they do mention, Tim, that this is suggesting to us that if there's a virus infection of the brain and the virus particles end up in the CSF, those could then end up in the lymph node where they're presented to T cells as part of the initial immune response.
Can viruses travel within just the lymph without also being inside cells?
I think so, yeah. I don't think they need to be inside cells, yeah. All right, next, does this matter? Does meningeal drainage make any difference in virus clearance or survival? So basically they ligate these vessels, and then they infect, and they find it makes the mice die more. Basically, it aggregates the mortality by ligating these vessels. They can also ablate them by a different approach, and they basically confirm the same observation, enhanced mortality, and they find more virus in the cerebellum and brainstem. There's more immune cells in there as well. There are more inflammatory cytokines. And so when you ligate these lymph vessels, you're apparently preventing virus from getting out of the brain, and you increase the burden in the brain, you increase mortality, you increase inflammation. So maybe it is a mechanism for getting virus particles out of the brain in addition to starting an immune response. Here's an interesting one. It's known that VEGF-C, we already said this is a factor in the lymphangiogenesis, right? They said previous reports have shown that treatment with recombinant VEGF-C can increase the diameter of lymph nodes and synergize with checkpoint blockade in treating brain tumors, right? Checkpoint blockade is a way to wake up T cells that have become tired of doing their jobs, basically, and this works together with VEGF. So what they did is they treated mice with recombinant VEGF-C, and then they infected them. They confirmed that the VEGF treatment increases the diameter of the lymphatics, and it improves the survival of the mice. There's reduced viral loads in the brain. So if you enlarge the lymph vessels, it has a good effect on virus infection. It ameliorates the severity of infection, which is weird, right? Because virus infection does it, but apparently the flow is impaired. But if you increase the vessels ahead of time, then you can improve the survival and reduce viral loads in the brain. So
yeah, they didn't really quite get into the mechanism of how that worked, unfortunately. They just show that if you make these lymphatic vessels get bigger and more, have more of them just before you infect the mouse with the virus, then the mouse survive better. But is it because it drains better, so the virus is detected more quickly, or is it because immune cells can get to the brain faster? Or what is it?
Maybe they just clear it out quicker, and don't have a chance to really infect cells. But I guess one interesting question is whether if there is clearance through the lymph nodes and there's an immune response. I mean, dogma would say that the antibodies, let's say, or any activated immune response wouldn't really do much in the brain. Or maybe it does. I don't know.
It depends whether the lymph vessel allow things to go from the lymph node back to the brain. They don't really talk about it. And I don't know how the valves work. Is it one way?
Yeah. No, I think you can regulate flow by the valves, right?
The other interesting experiment that I thought would have been nice for them to do would have been to do the VEGF-C treatment and then have a condition where they do that plus a ligation to show that like it's actually an on-target effect, right?
Yeah,
you don't know. I mean, VEGF-C could be doing something else is actually mediating the effect.
Yeah. So their idea is that maybe early in infection, these lymph pathways are intact, and then you do get viral antigen flowing to the cervical lymph nodes, and then you get an immune response. And they say maybe, the immune cells can migrate back through the lymph vessel into the brain to help terminate or clear infection. But we don't actually know if that's the case. Whether these MLVs are a tunnel for immune elimination is a good question. Something that could be addressed.
Yeah, it seems like you just do that, instead of injecting dye into the brain and look for it to show up in your lymph node in your neck, you just do the opposite. Yeah. But I don't think I've come across anyone doing that. But I didn't look at all very deeply into this.
So here's their summary. They've shown that MLVs can transport virus from the CNS to the cervical lymph nodes. VEGF treatment promotes lymphangiogenesis and helps with CNS infections. They say this suggests a therapeutic approach, but I'm not sure, right? Because they gave it ahead of time. I'm not sure if you can give it afterwards and it will make any difference. And even though the virus infection promotes lymphangiogenesis, the function of these is impaired. So there's a little bit of a quandary there, which I don't understand. But for me, it raises the possibility that viruses could come out. And if I were still doing research, that I would take poliovirus infection of mice, which we could readily do, and look at some of these questions. So one of the first things I did when I went to Columbia years ago was to make a transgenic mouse model for polio infection, and those mice still exist, and they could be used to look at some of these issues, whether the virus comes out again or not. So for me, this is a really cool investigation, which I think impacts what we understand about neurotropic virus infection. But I have to say one last thing. I always argue that, viruses getting in the brain is an accident. That doesn't help their dissemination to, the biggest driver of virus evolution, in my opinion, is transmission, right? Because if a virus doesn't get to a new host, it's finished. So you go in the brain, hey, that's a dead end, but this kind of suggests it might not be. So I really enjoyed that part.
Yeah.
So I have two questions. One is directly related, which is how would you go about figuring out if the virus itself is mediating the reduction in lymph flow? Like I was trying to think about how you could design that experiment, and it's actually quite challenging because in some ways you'd think that the virus would want a route out of the brain so they could get transmission, but on the other hand, you might imagine that the virus would want to decrease lymph flow so there's less immune sampling. So it's kind of an interesting double-edged sword for the virus.
It could be a host effect, right?
Yeah.
It may have nothing to do with it. May be some kind of immune response to infection to shut it down. I don't know. It's a good question. The problem is, Uri, that all of the viruses they use transmit really well by other routes. So
right.
It
so you
doesn't
don't
matter
really
if
need egress. Yeah.
You don't need egress, exactly.
Yeah.
However, here's the kicker. So people don't eat each other, but animals do. So you could imagine that getting in the brain of an animal is a good way to be transmitted because other animals will eat the brain, right?
So the other, I have a follow-up question, which is actually related to that, which is that, so if you think about, so HIV is a good example of a virus that now is thought to actually like have a reservoir in the CNS. So one issue with like chronic HIV patients is that they take antiretrovirals, they have no viral load, but they're not cured. And if you stop the antiretrovirals, then you actually have viral proliferation and they have like essentially recurrence of disease. And there's a lot of work, I think there's some work that suggests that perhaps one of the areas where the virus hides is in the CNS because it's immune privileged. And then this virus just sits there and then there's always virus leaving and it's cleared. It's essentially its replication is blocked by the antiretrovirals. And then if you stop it, then you have more egress. But this could actually be a mechanism for egress from the CNS. And I thought that was an interesting question that is like perhaps more clinically relevant in this case than pre-treatment of, for these acute viral infections. But what if you could increase lymph flow, get better sampling of the CNS space, and then have a more strong CNS-based immune response to HIV, right? Then you could actually improve HIV clearance in the CNS.
Yeah.
So I thought that was super interesting. But I like once in the past, in a long time in the past, went on a deep dive into this, and there's like pretty poor animal models. So I think it'd be very hard to test.
Yeah, I think it's a really good idea. I agree, but testing it is problematic because of lack of animal models.
Yeah. Because I also always wondered about that, where if it was restricted to the CNS, how would you get a proliferation afterwards? Or, how would it get escape?
Yeah.
Because because HIV is pretty neuron specific, right? Like there's latent, or is it microglia? I can't remember.
I think it's astrocytes was
astrocytes.
I think it's mostly non-neuronal cells.
Yeah.
And then there's so then essentially it escapes, it essentially proliferates, and then there's some sort of local viral production that then maybe infects endothelial cells and gets crosses the blood-brain barrier. I'm not sure, but this seems like a really great mechanism for it to leave the CNS, right?
Yeah, my understanding is that much of the HIV infection in the brain is non-productive, but it does induce the synthesis of cytokines that are toxic for neurons, right?
Right. So that's the other interesting point, which is that it's not just a reservoir when it's in the CNS, but as Vincent said, you can actually have long-term neurodegeneration and cognitive impairment from HIV disease that is independent of viral load in the blood. So just kind of the length from the initial infection predisposes to this cognitive impairment as opposed to the amount of time that you have a high viral load in the blood. And that is mediated by cytokine production and kind of like low-level CNS inflammation. The interesting thing, though, is that some of these mouse and rat models where they study HIV-associated neurodegeneration, they actually just express viral components in neurons. So it's like, they're not the most robust models. Yeah.
So, Ori, do you still see a lot of HIV-associated dementias?
I mean, in San Francisco, it's like quite prevalent, and there's actually a clinic here that specializes in management. It's like part of the Department of Neurology in the Memory and Aging Center. There's a couple of like senior people who essentially have built their career studying it, and it's super interesting because essentially like they get these kind of episodic worsening of neurologic symptoms. Sometimes they'll do a lumbar puncture and they see virus actually in the CSF, and then they can treat with CNS penetrant antiretrovirals and get improvement or like at least stabilization of their decline. So it's a super interesting entity. And in San Francisco, where there are a lot of people who have had HIV for a long time, the number is really high.
Huh. So then the thinking is that it's more of an acute viral effect versus, I mean, I know there was some evidence that you could get amyloid production and an actual sort of Alzheimer's-like pathology.
So I think that it's a mix. I think the data is like super poor for these like more like CNS penetrant antiretrovirals. I think but at that point they have like nothing to do, so they just will do it. And it's like, I think it's at the level of like case series as opposed to kind of randomized trials. But that's essentially the standard of care here, is that they transition their antiretroviral regimen to one that has better CNS penetration.
Right.
Yeah, and they'll track the viral titers in the CNS over time, in the CSF over time.
Right.
Yeah.
I think this is an important paper. I think it's going to have long-term implications in lots of viral CNS infections. So.
Is it possible to sample your cervical lymph node to see what virus has recently been around? Or I'm thinking I'm totally going on the
volunteer for that, but
no,
yeah,
I'm totally going on the New York sewage.
But the thing is, you don't know. There's probably 1 million viruses that are there that get cleared right away, right? Like it's just a sample. Like you don't know that that's caused some sort of infection or anything like that, right?
That's true. Yeah.
Plus, you'd have to biopsy it, Tim. Right? That's not everyone. I mean, some people have lymph node biopsies, but I think in general, it's not something people want to do. Right. All right. That'll do it for TWiN 33. You can find show notes at microbe.tv/TWiN. You can send questions or comments to TWiN at microbe.tv. If you enjoy our work, consider supporting us. Become a patron. You know, in the old days, the Medicis were patrons of the arts, right? You could be patrons of the sciences now.
My lab needs patrons.
Everybody,
I went to the Sistine Chapel.
Yeah.
Microbe.tv/contribute. Actually, Jason, we have done some appeals for lab funding to our listeners, and you can get a good response. But the problem is the amount of money you need to do lab work is orders of magnitude more than you can raise, So crowdfunding is not, in my opinion, all that useful.
No, you need to land some nice donors that have real money.
Yeah, you have to have a donor who wants to invest millions of dollars. Yeah. Anyway, if you want to invest far less, you can go to microbe.tv/contribute. Ori Lieberman is at UCSF. Ori Lieberman on Twitter. Thanks, Ori.
Thanks, guys. So we're almost at 3 years next month.
Oh, wow.
For the podcast. So
cool.
It's been a nice run.
Still going. It's great.
Yeah.
Jason Shepard is at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent. Yeah, 3 years. Wow. It's gone quickly.
Yeah, crazy, right?
I was in Ithaca recently, Jason, and I did a podcast with your old buddy, Cedric Fachat.
Oh, cool. Yeah. Yeah, we're still working. We're actually working on a paper right now.
We're
still
collaborating. Cool. Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent. Thanks, everyone. And great paper. I learned so much.
I'm Vincent Racaniello. You can find me at virology.ws. Been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.